Thanks, Jay. Yeah, I'm Greg Parr. I work with the USDA with Plant Protection and Quarantine out of Raleigh, North Carolina. And today we'll start off with an update on the Blue Marsh site in Pennsylvania. And presenting will be Dr. Rick Roush, who's the Dean of the College of Agriculture at Penn State. And then the second half will be Dr. David Biddinger, who's a tree fruit research entomologist at Penn State. So you can go ahead and start.
Thanks, Greg. It's really a pleasure to be here and to learn that there's so many people that are involved in managing spotted lanternfly. Somebody else has to stop sharing before I can, it looks like. Thank you. Okay, can everybody see that?
Yes. Yeah, yes. You're good.
Good, thanks, Rick. So Dave and I are going to talk about experiments we've done over the last couple of years in the summer at Blue Marsh, which is a large-scale woodland area where the woodlands are divided up in hedgerows around the reservoir area. And so they're long and narrow, typically about 15 to 20 meters deep and then continuous. And so it was a way for us to actually design this where we could have plots where we had relatively little risk of reinfestation from neighboring plots. So that was what drew us to Blue Marsh. In addition to the fact that when we first started going out there and looking at it in 2019, there were very high populations of spotted lanternfly. So the objectives we had in looking for a site like Blue Marsh were to try to figure out if there were ways that we could target woodland areas with control practices that would protect neighboring high value crops, particularly, or other horticultural goods, particularly grapes. So the notion was if we could come up with ways that would suppress them as they're developing in the woodlots that are neighboring grape vineyards and so forth, we wouldn't have this continuous over time fly-in of adult spotted lanternflies to vineyards that requires repeated sprays, and that at least we could suppress that some. We also wanted to see if we could use the kind of tactics to develop options that would increase the efficiency of control programs for containment and suppression, especially as we'd learned that populations seem to be moving along railroads, and you can often find high populations around railway switch yards, that this might provide a way, especially by the aerial application of controlling spotted lanternfly without being entangled with the problems of access and safety around the rail yards themselves. So we wanted to look at aerial application in particular. We were following up this research with, after success at a trial in the Norristown Farm Park near Philadelphia with Bavaria bassiana, where we did a very early trial based on when I saw some of the results produced by Eric Clifton and Ann Hyack from Cornell. We agreed to try moving directly to a large field trial with Bavaria. In this area, we had a relatively small number of replicates and went with split plot design because we just had two treatments, the treatment and control. And we were able to demonstrate about 50% suppression of nymphs in our trial, which encouraged us to try it again at Blue Marsh. We also wanted to test the impact of the various treatments, Bavaria and Dinotephron, on non-target species, which David Biddinger is a particular strength of David Biddinger and his interest in it. And what you'll see probably is the largest data set collected on any kind of insecticide trial that I've ever heard of. So we were looking at the impacts on non-targets, and we also wanted to use the amount of data we were generating to test and compare two different sampling methods. One, the use of sticky traps or sticky cards on trees, and the other, time visual accounts, which is what we were forced to use at Norristown. Typically, these visual accounts involve at least 20 person minutes per plot, preferably at least 30. And what we wanted to find out, it was whether these gave you data that was at least as accurate as you could get from sticky cards. One thing about them is you don't have to set them up ahead of time. You can count and spray on the same day and so forth. So the experimental designs we used at Blue Marsh over a couple of years included a completely random design in the first year, and then, for reasons I'll explain later, a block design in 2021. And these are, for us, were relatively large. We would probably have liked to go larger still, but even though the Blue Marsh area is quite expansive, there weren't so many areas that had what looked like a relatively uniform population of tree of heaven, and therefore we thought a relatively uniform population of nymphs. So we looked at Bavaria and Dinotephron in 2020. We picked Dinotephron because the experience that both David and I had with pyrethroids compared to neonics in terms of their persistence and impacts on non-targets. And although many neonics are rightfully being excoriated over their impacts on pollinators, what we knew about dinotefuran is in principle it could be a bit different, that especially with sprays, the residue impacts seem to last for something on the order of a week or so, not a very long time. We also thought it would have advantages of doing a bit of, since it's it can translocate in the plants, that it might be able to get better coverage effectively for the nymphs. In 2021, we tried to compare dinotefuran and bifenthrin in terms of their efficacy for control, and also look at their impacts on non-targets. So we had several treatments. We typically were running about 45 to 50 plots. We had 6 or 7 replicates, which we thought would be big enough to counter any variation in the plots. That proved to be wrong. The applications were timed to specific inscars, as I'll show you in a moment. So we were looking for nymph and adult population densities and comparing sticky cards and time visual counts I mentioned earlier. So in the first trial, led by Dennis Calvin and his work on looking at day degree accumulation models for hatch and so forth for spotted lanternfly, we managed to time the application so we were very close to 100% hatch. And we were also, so what we were trying to do is target the first and second instars. And also this is a time of the season where there aren't so many flowering plants available for the pollinators. What we found when we looked at this with sticky traps was within a few days after the trial, a few days of the applications of the 15th or 16th of June, you could get roughly 90% control. And then we saw this kind of level of control persisted for weeks, except in the plots that were treated from ground, where it seemed to diminish. At first, we thought this showed a real advantage of the aerial sprays until Dennis went back and carefully looked at the geographic alignment of the plots. And we realized that by chance, more of the ground treatments were close to places where they could have reinfestation from outside the plot. So what we think was happening is that over a period of time, the plots were, some of these ground treated plots were reinfested. In fact, we went out and tried doing some tests where we took nymphs from control plots and caged them on treated plots to see that had been treated from the ground or from the air to see if the residues had declined. And the results were sort of mixed, but suggested that probably after a couple of weeks, there was very little residue activity left and nothing to stop in-migrating insects. Nonetheless, we take this as, if you consider this from the standpoint of the size of the plots, if the aerial application was over quite a large area, you wouldn't expect this reinfestation to occur, and that either control tactic would give you something on the order of 90% control at that time of the year anyhow. We also looked at this for visual counts. And the problem with visual counts is you really can't start to execute them until you have a significant number of second and third instars. It's just too hard to find the first. But in a very parallel fashion, the results were essentially the same between the sticky cards and the visual counts. You got roughly about 90% mortality from the safari air applications. We looked at them within a week or 2 weeks later. You had somewhat less in the ground applications. But again, we think that's probably, by the time we could go back out and do the counts for the nymphs, there would have been ample opportunity for in-migration, confounding the effects on the ground plots. So in short, the Blue Marsh, we showed that safari applied by air reduced the nymph populations significantly and could be a very useful tactic, particularly for controlling populations along areas like rail yards. The ground, we think, probably works just as well as long as the plot size is very large, so you're not getting reinfestation. And I'm foreshadowing some of what David will talk about, is that we put out pitfall traps and a couple of aerial traps and could find no significant impact of the dinotefuran on non-targets. We also collected a lot of data on non-targets for Bavaria, as well as trying to look at the impacts of Bavaria on controlling the pests. And to our great chagrin, despite a lot of investment in this and spraying the plots 3 times with Bavaria, we were unable to demonstrate control by Bavaria in these sites. Part of it was there's some variation because the plots were more variable than we anticipated, but also it seemed that in contrast to Norristown, where you had a very specific design where you could compare things side by side. But it was also, it's very much a wake-up call that that it's not as robust a treatment, and it's harder to demonstrate continued efficacy with Bavaria than it is with other insecticides. In 2021, we set out to do a much more detailed comparison of bifenthrin and Dinotefron, looking at later in the season when you're starting to target third, maybe fourth instar nymphs, but also adults. This on the top left, you see kind of an aerial depiction of what Blue Marsh looks like, and you can see all the hedgerows and so forth that we were working in. And we tried to use ultra low volume with a mist sprayer. We decided to test out a different way of applying these than by the hydraulic sprayers. We had some trouble on July 8th. It was this photos were taken actually in August, but on July 8th, it was a bit of a rainy day. There was a light rainfall, and we had some other issues in terms of getting the spray on. And as a consequence, I was decided to be careful in terms of the counting, and we decided to do the counts at both ends of the plot. That is to say, sides of the plot that were closest to where the sprayer was, and ones that were more distant, because I had some concerns about how much penetration we were really getting back to the back of the plot. So in fact, that's what seemed to happen. We've got residue data too that I've skipped just for the sake of brevity, but this is what we saw in the plots, and you can see Safari. If you look at these, are just the samples looking at near where the sprayer was, and what you can see is Safari looked like it was pretty effective, at least on some dates compared to control plots, but not always. So in the 12th of July, Safari looked pretty good. Data from the 15th of July, frustrating and not so good. Better in the 19th and 26th. But notably, Bifenture was pretty effective all the way through this time period. One of the things we think could be happening is if you apply Safari late in the season, it may not be translocating through plants as efficiently as it was during the early part of the season. But this is what happened if you look at the samples on the far side of the plots. And here, the vulnerability of Safari not to work as well is much more distinctive, as you can see. In a lot of the circumstances, it's not, especially just shortly after the spray, it wasn't any better than the control. But even with Bifenture, you could see that we were getting less control than we got at the near side of plots. And it really was a wake-up call that the sprayer was not really getting even 10 meters deep into the plots very effectively. So we tried it again on the 19th of August, where we hope to get more data about late instar nymphs and adults. And again, you can see much the same sort of thing happen, that Safari, the Dinotefron, was not consistently very effective through the plots, and Bifenture was much more effective. And it was, if you looked at the data from the far side of the plots, it was really poor efficacy across the board for Dinotefron in particular. So in summary, we've demonstrated, we're pretty convinced that Dinotefron can suppress the nymphs by at least 90%, especially if you target the early nymphs, which would reduce adult pressure in neighboring vineyards. So this could be something that could be applied by people locally. For a lot of the USDA and state programs, you need your workers working all season long. And so later in the season, Bifenthrin might be a better choice, but early in the season, before the pollinators start flying effectively, we think this is a good tactic for people who want to try to protect grape vineyards. The aerial applications, Dinotefron, would seem to be very good suppression with minimal target effects to target areas, again, like railroad tracks, where you really want to drive the population down, have limited access, but you want to be careful also not to do too much damage to non-targets. I know there's a lot of interest in Bavaria still, but as a person who was the first proponent of getting out in the field, I have to confess that it doesn't look like it'll be reliable. In contrast, Bifenthrin does look like it's pretty robust for control in the later part of the season. And the real question we had was about non-target impacts, and David will address that. And we've discovered that time visual sampling was at least as effective at looking at sticky cards, and also that we ended up going with blocked experimental designs to do a better job of controlling the variation between plots. That is to say, when you're looking at egg hatch, it's really hard to go out and do pre-treatment samples to assign blocks. But if you wait till you start to get second, third instars, a good strategy is go out, do counts, figure out which of your plots have high and low populations, block them by population density, and randomly assign in those among those blocks. That from the experiments we ran, but especially the statistical analysis done by Dennis Calvin and Joe Keller, suggested it was a much better experimental design in the fields. So with that, I'm happy to pass it on to David.
Thanks, Rick.
Sure.
Thanks, Rick. One of the things that we've done up to this point too is that, for 3 years, we've tested on potted plants, the grapes, peaches, and tree of heaven, different insecticides, looking at efficacy on both the nymphs and the adults, the residual activity, no one came in. And when we started this in the field, Rick talked about the Bavaria. I'm a tree fruit entomologist for 30 years. And when the choices come, and I work a lot with Xerces on bee effects from neonics. So when the choices come down to neonicotinoid and a pyrethroid, the IPM person to me shrinks at the thought of putting a pyrethroid out there because of the long non-target effects that it has in orchards. And it disrupts all the biocontrol, will kill ladybird beetles for like 2 or 3 months after a single application. The neonics we have to worry about because, of course, it can move systemically into the nectar and pollen. But we've done work at the station here looking at fall, August to early September applications of dinotefron on apples, because we use it for Section 18 for control of brown stink bug, another invasive pest that's very similar. And we do not see with the foliar applications the carryover from one season to the next. We do see that when we use trunk injections. I have a student working on that in red maples and tree of heaven. So the concern with the neonics is the impact on bees. And I basically have worked out 3 different methods to try to evaluate the non-targets. And the one that's probably the most targeted for the bees is blue vein traps. And there's a picture of one there. They're very good at monitoring all types of pollinators, especially the bumblebees and some of the larger bees, not so much the parasitic ones, and some of the parasites like this Staphyid wasp that's the main biocontrol agent for Japanese beetle. It's very time consuming. While we were doing this, keeping in mind that although I love looking through tons and tons of insects, we want to come up with the idea of bioindicators where we can simplify the system so that PDA or USDA can do a simplified version of this and figure out only a few taxa. I think, I estimated just in the blue vein traps, I've done over 75,000 bees and other things closely related over the last couple of years. So it is a large data set and is very time consuming. The sampling is not the hard part. The hard part is getting them labeled and identified. That's where it takes up a lot of the time. Rick, go on to the next one. And in Blue Marsh, there's over 135 different taxa, and mostly of bees that we were looking at. So these are some of the groups that would come to it. And the idea, and we monitored all of them, kept track of all the different numbers, but really we're looking mostly at the bees to see if there's any bee effects. Go ahead. And you see here, I don't want to confuse people with the other. We got tons of data tables, but you see effects like in the second one, the bombas combined, which is all the bumblebees together, where on week 2 or the week just before, you get these transient effects where soon after the application, you'll get a reduction, or it may take 2 or 3 weeks as the larvae are feeding on contaminated pollen or something like that and finish development. We won't see the community be a reduction in a certain types of bees. So it can be really transient, we can be like in the very first table there where we look at it over time. This is basically multiple counts up to 20 counts, and we summarize the area under the curve. It's an old trick that they use for orchards and plant pathologists. So we can do the ANOVAs on each date, but we also get this cumulative effect that we have on the other. And it's usually the cumulative effect that shows the differences, the red there that Rick's pointing at. Of these groups, the Bavaria trials, basically, there's a lot of literature out there saying they're very broad spectrum, but our experience was Bavaria in both years, it had no non-target effects in in 2020, and it tested in 2021. So we can just put that out of there. Bavaria may not be the most effective, but it's also we've not been able to detect any impacts that it has. The bees that we're really concentrating on were the bumblebees because they're charismatic and the problem with the plots being a little bit small. But there was one species, Bombus bimaculatus, which tends to nest in the wooded areas where we have the plots, that was affected over time. You can see there was a big reduction in the bifenthrin, an average of 4 days over, basically significantly different. But that was for the first spray for the nymphs. The second spray, the problem with combining all bumblebees together is there's no Bombus bimaculatus, that particular species, in the second half of the season. But there are other species that kick in. So that's part of the reason why you got to get some species level IDs. You just can't group them. Of the ones that we've looked at, the groups at the bottom there, these are the groups that we think in a forested situation would be the ones to monitor. The Ceratina, which is a small carpenter bee, it nests in raspberry brush, and it does not fly very far because it's really, really small. those are going to be localized. This Malaise bimoculatus, which is a really black bee with white legs, is very distinctive and easy for anybody to identify, is another one that we have seen some transient effects on. And then this green bee, and there's a lot of other metallic green bees, is Lagochlorpura, nest only in rotting wood in the plots. So you know it's not flying in like the bumblebees are from the grasslands next to it or the crop next to it. They're coming from the plots. And those are the ones, you know, if I had to separate the 135 down, I could probably put it down to those 4 or 5 taxa and train people how to look at them. Next one, right? Okay, the other method, it's less time consuming, and it's based on kind of a neat little concept that Rick and I both think is kind of neat. San Jose scale, it's pretty much ubiquitous. It's on a lot of different plants. It's a real serious pest in orchards, and we've done a lot of work where we use the pheromone trap, where it'll bring in the males of the San Jose scale, because we've had a lot of anecdotal stuff, and we've seen in orchards, whenever you use a pyrethroid, you flare up scale insects because you're killing off the biological control agents. Same thing with mites. But this is a very good way of putting up a pheromone on a small trap, looking it over for both the parasitoid and the pest that it may flare up, and be able to establish what's going on with both of them. And we basically found that bifenthrin can affect both the scale insect and the parasitoid. Dinotafron really is not causing problems. And a large part of that is because bifenthrin, like I said, will last as a foliar application with surface residues for certain biological control agents and spiders for like 2 or 3 months. But according to the USDA environmental assessment, the Forestry Service, dinotafron and a lot of these things, they're basically absorbed into the leaf or the plant within 36 hours. So it's a very short pulse when they can be available by contact. So after that, it's just things that feed on the plant itself. Go ahead. I looked at a lot of different groups. I don't expect people to be able to go through all this. The little yellow orangish wasp on the left there is the parasitoid of San Jose scale, Carcinosis. It's very distinctive. It shows up on actually in higher numbers than the San Jose scale does itself, but we have seen reductions with both dinotafron and bifenthrin, but the effects tend to be stronger and last longer with the bifenthrin, which kind of goes in with our experience. While I was looking at these sticky cards, I just happened to look at all the other parasite Hymenoptera, the Chalcidoidea, which is the middle group there of the tiny little wasp. And that one is also greatly reduced. And a lot of insect populations are monitored by these tiny little wasps. The other thing that popped out of all the stuff we looked at is bark lice over there on the right, that one is very heavily impacted by bifenthrin and somewhat by dinotafron. Go ahead. Okay, the other method, we had all these good things going on. It was, weekly counts on all these, and we tried to break it up before and after the first spray and before and after the second spray. And again, there, out of all the different taxa we looked at, there's the ones you see pictures of are the ones that were really the ones affected. The thing when people think about ground fauna is basically crab beetles. And in none of our treatments, did we really see an effect by Bavaria, dinotafron, or Bavaria on ground beetles, which is encouraging. There was some reductions of pill bugs, which are little isopods there. And we've seen probably the best bioindicator are actually spiders and daddy longlegs, which are the Opiliones on there. The little black bug down at the bottom there is called a burrowing bug. That actually feeds on plant roots, and we have seen a reduction in that, and they will come into the pitfall trap. So that's another one that we can monitor. The effects on ants have been minimal. Sometimes I think a lot of it's just due to variability in the plots, whether there's an ant nest or not an ant nest there. But that's another one that's easy to identify and you can monitor. And we have seen some transient reductions, like that very first table there, looking at the ants, the 17.5. I always say I can't even read that myself. But yeah, there's been some significant reductions in the case of bifenthrin. Go ahead. So the pitfall traps, like I said, those are the groups I would look at as bioindicators. And in all these cases, I don't think anybody other than myself, actually even myself, doesn't want to go through looking through all of these insects for weeks and weeks on end. If we can, blue vein traps in particular, they're like a gallon-sized container. If we could throw out 99% of it, that it really has no meanings, and cue in on these bioindicators, that's what we're trying to do next, is to be able to train people to come up with kind of really distinctive groups to see what the effects are of the different treatments out there in the woods. And slugs actually were surprisingly a good indicator in a lot of cases as well, especially with dinotephron. We didn't have the problem with the data from last year, 2021, was like what Rick indicated with coverage. The data is kind of all over the place. I want to repeat that again this year, even if there's not spotted lanternfly on plots that haven't been treated before, just to cue in on the non-target stuff. Now that we know what's out there, whether these bioindicator groups, and have complete coverage. So we're thinking to maybe go back to these plots and apply either handgun, which gets pretty good coverage across the whole thickness of the plot, or by going with these mist sprayers from both sides, just so we ensure we have complete coverage. Go ahead, Rick. So the data that Rick was referring to with the UV sprayer, basically our residue data said it only reached about halfway through those plots in 2021. We think we had really good coverage of the aerial applications. We think we did really well with a handgun, but the mist sprayer, especially was set up, there was some confusion about the rates according to the manufacturer, and it just didn't penetrate as far as they thought. So our non-target data from 2021 to me is a little bit suspect. The data is not as clear as it should be, so we want to give that another shot. So basically the summary is foliar applications of dinotephron, despite all of the bad hype and the impacts on bees that you see out there, when it's a foliar application, it's a short pulse that disappears very quickly from the surface of the leaves and only things that feed on it. And we've actually developed phenology models of tree bloom of probably about 100, 120 species of plants out there, so we can make sure we avoid applications before basswood bloomed or tulip tree or something like that that bees like to go to. So what Rick referred to as that July application for the second, third instar nymphs turns out to be a really good time to apply them, because you avoid the bloom of a lot of other trees that are out there at the same time. I know it's hard to time your sprays when you're covering thousands of acres and stuff, but in IPM, we try to choose the most selective compound at the most selective time. And if you do that, we should have minimal impacts. And like Rick also alluded to, we've seen better efficacy on dinotephron on nymphs. And that's at the time when there's a lot more bees and non-targets out there than in the fall, where the bifenthrin would knock them out. And also dinotephron in the fall doesn't work as well, we think, because it's moving, the sap's moving back into the roots. So dinotephron is not working as well. There are some problems too, I think, with dinotephron in a really rainy season, that it may flush the dinotephron out of the trees. So weather might affect that more than bifenthrin. I think bifenthrin's probably a little more robust, a little harder to, it's a little more idiot proof in terms of timing, but you're going to pay for it by maybe flaring up secondary pests like San Jose scale and wiping out more non-targets. One thing we didn't get good data on last year, we tried to look at non-target leps. We put gypsy moth, phenomon, and the residues that came out of there, but the results were all over the place because of the coverage. But a big concern is, bifenthrin being a surface residue and maybe potentially lasting several months, is it going to kill a lot of non-target moths and butterflies that are out there? And the other thing that came out of the residue stuff is that we did get drift. It was in the neighborhood of like 5 to 10 parts per billion outside of the plots and beyond the plots. And this is a real concern when you compare dinotephron to bifenthrin for fish toxicity, because bifenthrin is 1,000 times more toxic to fish than dinotephron. So not just the plots we're spraying, but the drift and the impact outside of that area. We need to try to minimize that. Did you have any other comments, Rick?
No, I think you covered it well. Thank you, David.
Okay.
So I think, Jay, we're just out of time, but we'll be happy to try to field questions by chat or whatever, by Q&A or whatever.
Yeah, if there's anything, I don't see anything right now in the Q&A, but if there has been some discussion in the chat. So yeah, if you would definitely go in there if you want to address any of that or comment on that, that'd be great. But yeah, thanks for the presentation. That was excellent. Trying to keep up with the agenda. So we'll be moving on to the next session, the next topic, I'm sorry, which is monitor and detection. So our first presenter will be Joe Francese, who is with our USDA Forest Pest Methods Lab up in Massachusetts. And then I'll introduce Joe Keller once Joe Francese is done.
Thanks, Greg.
Yeah.
I'm just gonna go here. Sorry. Okay. Hi. So I just wanted to I'm going to be talking about some of the work that we've been doing this year to improve survey methods for SLF, and this is work being done by our crew at the Forest Pest Methods Lab, formerly the Otis Lab, and we're this is work in my group, Miriam's group, and with Damon Crook, who's also at our lab. So this work, I think I've shown this slide, these thank you slides so many times. Our work doesn't get done in a vacuum. We have so many people that help us with all of the work that we're doing. So I need to send a shout out to everybody who's been helping us these last few years. Just to give you a little primer on what we know about SLF trapping. We've been working, we've kind of been working with different traps that you can put on trees, and we found that traditional sticky bands just really aren't as effective of a tool as we found that bug barrier, and more recently we found circle traps, which are traps that work on the tree. They're not sticky, and they catch them in a collection device, are ideal for, or not ideal, but are the best tool we have right now for trapping SLF. We've tried canopy traps to catch flying insects, and they really don't work. So just a little more history. We found that the bag traps, especially the circle traps fitted with bags, do better than jar traps, and both in trap catch and in detection rates. So our ability just to catch one insect at a site, doesn't matter how many we catch, as long as we can catch that one on a trap, we consider that a detection, and that kind of helps us determine how good our trap is at just finding that one SLF in a haystack. So just kind of my evolution of the trap to where we are now, and so we have this, and this bag trap is really the one I'm mostly going to be talking about today. So this year, we had a couple questions we want to answer. How do circle traps do on black walnut? Can we use black walnut as an alternate host when tree of heaven isn't available? Some of our tree of heaven doesn't grow in places where you can get to it, but maybe you have black walnut that's kind of close. We got a lot of questions about pesticide strips last year and aerial Malaise traps, so we wanted to look at that. We also looked at check frequency, looking at extending our check frequency. So I think right now the time that the program uses is about a 2 to 3 week check cycle, and we want to see if we can push that up to 4 weeks or even 8 weeks. And then the last thing I'm going to talk about is work we've been doing with Damon with color and LED traps. So, and I'll talk about where we're going next year. So we chose black walnut this year just because it seems to be a good host just to start. We wanted to know how will it work when compared with tree of heaven, and we kind of looked at some of the research that's been done by Miriam Cooperband's crew in Pennsylvania and here in Buzzards Bay, and then so we had our traps within our replicate were up to 150 meters apart, but we really tried to get them closer if we could. We had 38 replicates that we placed in 5 states. So like I said, a lot of help from different departments of agriculture, from PPQ, from different groups, and I'm really grateful for it. And we're hopefully those traps could be put in low density sites. And again, I'm just going to give my definition of SLF is one in a trap. So we put out 38 replicates. We had detections in 26 of those 38 replicates. And overall, our adult SLF trap catch was higher on tree of heaven. As you can see, there wasn't any significant difference between nymphs on tree of heaven or black walnut, but the adults really did outcatch, were outcaught on tree of heaven, but there wasn't any significant difference between species at any stage in detection. So you put a trap out, you're still able to get that one SLF. If there's SLF there, we were able to find it on the tree of heaven or on the black walnut, and we were able to get it in our traps, and that was at all life stages. There wasn't any significant difference between any of the life stages. And even when we went to low and medium density sites, we found similar results. So finding those sites, you get less and less because when you're setting these sites up, you're not really sure what your density might be. But and so black walnut does look to be a suitable host. We're going to look at maple next year as another host for traps, and compare it with Tree of Heaven in some of these outlying areas outside of the main infestation area, where wherever we can get them. And we'll be looking at the genus. I think we're gonna focus on silver, sugar, and red, but it's what folks have. So one of the questions that came up last year was, is the Dichloros pesticide strip necessary for trap catch, and do we actually need to have it in the trap? I know there's some questions about pesticide use, and not everybody can do this in every location when they have, handling these strips is not always ideal. So we did trapping assay, strip and no strip, and insects were placed in the bags beforehand, and then we also collected insects as they were coming into the traps. There were 4 collection periods. They were they ended in June, beginning of June, end of June, middle of July, and beginning of September, basically to coincide with some different life stages. July being our late instar, and then adults in September. And what we found was that there isn't any significant, across all the life stages, there isn't any significant difference between the number of SLF we collected or that we retained in our traps, with or without pesticide strips. So good news is that at least for surveys, you probably don't need to be using them. We might continue to use them a little bit from our research, but I don't think that, I think that you can forego the strip if you need to. So another question that we got actually at the summit last year, I think I heard the question a couple of times, where people were asking about aerial malaise traps and how those would work, flying intercept traps. So we had been trying the intercept panel traps that are used for wood boring beetles previously, and we found they didn't work. But we found these traps. They were from BioQuip. They were kind of expensive. They were about $273. We added our bags that we use in the circle traps to the top funnel. At the time, the lower funnels weren't available, so we didn't go with them. But we figured that was probably okay, because we know that SLF is probably going to go up the trap and into the bag more than it would go down into a funnel in the bottom. So we compared this with the circle trap, and I just have their prices there. So you put a circle trap out, it costs about $18 for the season. This BioQuip trap cost about $273. We did 5 replicates, and this is Sarah, who works in my lab, and this is pretty much what every bag trap looked like, or every not circle trap, but every aerial malaise trap looked like. We would have a few SLF on the outside of the trap, and then a bag of water inside the trap. They also were kites, and so we had to stop the study early because we had a nor'easter that came through, and I'm glad that we got the traps out of the field before that nor'easter came in, because I think that the landowner would have been kind of upset with us because our traps would have wound up somewhere else. We caught very few insects in malaise traps, but we caught hundreds of SLF adults in our circle traps. So this didn't really work for us, but we gave it a try. So another study that we worked on is our trap frequency check. We wanted to know, can you really expand on the amount of time that you have between checks? So could you put traps out at, right now they're being put out at about 2 weeks, but we're looking at 4 weeks and 8 weeks. And we're getting some processing help from Xavier. It doesn't look like there's a difference. So, and based on conditions of samples and experiences from what we know, and I'll talk about this I'll show you these traps in a minute, but we had some vandalism, we had fallen traps. So leaving traps out for 2 months in a field season, in a 6-month field season, isn't going to work. You kind of have to get out there. You kind of have to check on them, make sure that they're still up. So if you need to go out to 4 weeks, they're not that decomposed. They're not in that bad a shape. They're still okay. But yeah, and we found that our vandals came in all shapes and sizes. So these guys came through one of our color studies and took down a couple of our reps. So we just know from experience that we are we need to be out there and have eyes on the traps a little bit more. So the last thing I'm going to talk about are visual attractants for SLF, and I think I touched on this last year. Damon Crook in our lab had done some electroretinogram assays with adults in late 2020, early 2021, and he had found that there were adult responses in this 460 to 500 nanometer range. They were a little bit higher than other areas. He wasn't able to work with earlier instars. The prep was not as easy, and I can go into this more, but this is the basic gist of how you basically flash a light over the retina, and then you get and you record your responses for each insect. So he was able to get his hands on some adults and run the retinogram. So he had these ranges, and what we did was with that we took we went out and found 6 paints that it's a paint and primer in one that you could find at Home Depot. We just picked out some colors that seemed that looked like they were going to match in there. And if anybody has seen our EAB stuff in the past, we kind of use this process again. And we measured the peak wavelength and the reflectance of each paint, and then we chose 3 paints. And so these were all available. Like I said, we went into Home Depot and we were able to find these. And so you had these 3 colors that were in the 490 to 460 range, based on what's available. And we went with spray paints because figured spray paints would be easier to deal with on circle traps. We sprayed the circle traps with the paints. We also had an unpainted circle trap control. We were able to set up 15 replicates. Each color was deployed in a block on adjacent or near adjacent tree of heaven of comparable DBH. And so we tried to keep it within 5 centimeters for each tree. And then the block was close. It was as close as the trees could be. The trees were as close as they could be. Our initial placement coincided with the expected beginning of adult flight. So I think it was July 12th when we went out there to start this. And then traps were checked every 3 weeks until the end of October. Unfortunately, we didn't see any differences in our numbers of adults caught or nymphs. Because we got out there later, we didn't really see nymph emergence. We didn't have a lot of nymphs to catch, but we did see that there might have been some differences, at least in the 4ths and maybe the 3rds for the blues. So that might be promising maybe this year. The circle traps, we wouldn't be using the circle traps. We'd probably want to use a sticky trap, and I'll talk about that in a second. So there's no significant difference between our control and our painted traps. All the trap catch was male skewed this year, and our 2 of the colors did catch up at the end. Our trap catch was really low on them until the last period of flight, but it was really at the end. And I don't think that I'm not sure that anything was going on there other than those traps were just doing a little bit better at the end. Problem with leaving the traps up in a high density area is though that all of the traps did start to turn black by the end of the season. The last thing I'm going to talk about is our LED trapping. So again, we took some of that, took the retinogram results, and we went out and tried to find a blue light. So we found these blue and white powered LED strings. You put batteries in them. They're for indoor outdoor use. 3 AAAs. They're attached to trees with standard circle traps. You can see we kind of made a little made a wrap around the tree, and then we had a basically a landing strip that went straight up into the trap. This thing's right up on the wooden support of the trap. Lights have an 8 on, 16 off timer. So in the morning, when we were setting up, we turned them on. We used a blinking setting to conserve energy. And so here you go. You've got a white trap here, a blue trap, and then an uncolored trap, an unlit trap. We did 15 reps. The batteries were changed every 3 weeks. We had a major problem at 6 weeks, and our batteries that we had bought all corroded and all the lights had failed. So we had to actually replace all of the lights, and that took us a little while to do. We're in the process still of counting these samples. We had a few 100,000. We had about 100,000 SLF that we caught this year. So we're still going through samples, but that doesn't seem to be different. Doesn't seem to be a difference among any of the treatments. We were able to catch a little bit at the beginning of flight and a little bit at the end, but for the most part, it was the same number of insects and traps. So they were just going into the traps. So final conclusions for this year and plans for next year. Black walnut has the potential as an alternate host of if you can't get the tree of heaven, you can't access it, but it's near there. You can use black walnut. We'll continue that this year with maple. You can remove the pesticide ships from the protocol. It shouldn't affect trap efficacy. We think the traps will keep working. This year we'll probably do some work with trap density, increasing the number of traps in close proximity, and seeing if there's anything that we can do to figure out some if also population density studies using traps. So the last thing, the paint colors and the LEDs that we tested didn't increase catch over control traps. We are planning to conduct some earlier trapping this year in the early parts of the spring and summer to determine if the colors affect nymphs. We would probably use sticky cards just because they seem to work. Sticky cards do seem to work better for the earlier nymphs. And the other thing that Damon and Miriam and I have kind of batted around just a little bit after some talks about a month ago is looking at some other visual attractants and other visual cues. Might be, some questions have come up, and there might be some other visual cues that we might want to look at. The patterns on the wings and other insect patterns that might be worth trying to check somehow. So with that, I'm done. If anybody has any questions, or I can go to the Q&A, Greg, so that Joe has more time. So yeah.
Yeah, thanks, Joe. Yeah, there's just a couple in there. I think Dean Roush answered some on that, because there are some questions about a lure or attractant, but yeah, there's not one currely.
Yeah,
there was another one. Yeah.
We did use we did use the we did use methyl salicylate lures on all of our traps, especially in the with the black walnut, just because we don't think it would work as well with Tree of Heaven, just because it's the same compounds that are being picked up by Tree of Heaven, but it might affect. The thought was we might increase our catch on the black walnut, and I forgot to mention that. Yeah.
Okay, thanks, Joe. And then, is there any plans to test the circle trap catch on China berry?
That could be something that we could do in the future, because we're going out to these sites that are outside of the general area. We're picking one trap at a time. We're picking one tree at a time, just because we have a lot of people that are helping us. So maybe that would be something that we could do in the future. Yeah.
Okay.
Yeah. And then and Julie's, yeah, we get there. Yeah, that trap was gross. I don't know if you saw it. It wasn't out there long enough, but
about the city mold. That's a question you're answering. About city mold on the Malaise trap. Okay.
Yeah. So. Okay. Thanks.
Yeah, and there's a couple others in there, Joe. If you can get to those,
I'll see what I can.
Great.
Yeah.
All right.
Okay. Thanks,
Joe. Yeah. All right. So then we'll move on to Joe Keller, who's a postdoc working with Kelly Hoover at Penn State. Whenever you're ready, Joe.
Okay, let's see if this will go. Are you seeing the presentation?
Yeah,
okay, awesome.
Looks good.
Thanks. Yeah, today I'll talk about monitoring spotted lanternflies and some lessons we learned looking at outlying populations of lanternflies in Pennsylvania. So a quick outline of what I'll hope to touch on. First, a look at the locations where we've been doing our sampling at these outlying populations. Then talk about what we've learned about doing egg mass counts, time visual search as a method for measuring lanternflies and detecting them, and traps, the same traps that Joe just talked about, the circle traps and bug barrier sticky bands, and then touch on some future directions at the end there. So we know from the history of the spread of lanternfly in Pennsylvania that these long distance dispersal events can create isolated outlying populations. So you get these cases where you see what looks like a new island of invasion, basically, where you've got a population showing up where there's nothing exactly nearby. And so we've focused our sampling at these sites, hoping to learn some things about lanternflies. They're an attractive study site for testing surveillance methods because you hope, and I think generally we found across the sites that we've looked at, there's predictable variation in lanternfly densities. So you get a higher density core where the population first started, and then as they spread, you get to lower densities further away from that core. They're kind of a window back in time to early invasion, so what happens when the lanternflies get somewhere new. And they're also interesting to us as a place to assess treatment, whether treatment can locally eradicate lanternflies in some of these spots. So we've sampled at 5 different towns across Pennsylvania. First in Lewistown, where we've got 7 plots. We've added 7 more this past winter, trying to get some higher sampling density there in Lewistown. But yeah, 7 plots in Lewistown. Johnstown, again, we've got 7 plots and sampled just this past year, kind of spread out there in Johnstown. And these plots are a little bit opportunistically placed. So they're kind of in edgy habitats where we can get frequent access to check our traps, basically. So they're not systematically arranged plots for these, but they're places where there are a lot of host plants for lanternflies tend to grow at these edgy habitats. Huntingdon, we've got 11 plots, kind of range town there. Shippensburg, 16 plots. And then Altoona, we've got the bulk of our sampling. We've got 31 plots that we monitored in 2021. And they're kind of arranged along across the whole town there. So first I'll talk about what we've learned about egg mass sampling from these plots. So each of these plots is a 20 by 5 meter plot where we go through and measure the DBH and identify all stems in the plot bigger than an inch across, bigger than 2.5 centimeters across. And we identified them if we were able to. I'm not the best forester, so we get Tree of Heaven for sure, sumac and black cherry and maple pretty well, but a lot of trees go into the unidentified category for us. But then for each tree, we count the egg masses present on the tree, and we make a distinction between new and old egg masses, and I'll have a picture of an old egg mass later. We also distinguish between egg masses above and below 3 meters high on the plant. And in addition to searching every stem, we search other surfaces. So we're looking on rocks or fallen branches in these plots too, where we do find eggs laid on those other surfaces. We use binoculars to aid our search over 3 meters. So we let the observer search from at least 2 angles for every tree, but we don't limit the number of angles that they're allowed to look at. So we let them wander around and look over the whole surface of the tree. And generally we found that you get pretty good coverage from standing close to the base of the tree and looking straight up, because the eggs are frequently laid on the underside of things. And we take care to avoid false positives. I'll have a picture here of one lookalike. You do get a lot of things that kind of look like egg masses if you're not paying close attention, especially over 3 meters. It can be trickier to distinguish. But yeah, we're trying to be as careful as we can to avoid false positives. And we did this for eggs that have been laid in 2019, 2020. And you can see here's an example of, you do kind of have to get down on the ground and look around for eggs, because they tuck them away in these little sheltered areas. So this is the underside of some bark of black cherry trees, I think. So they're tucked in there. So this is a look through binoculars, kind of showing what you're up against looking at the surface. We use 8 times magnification binoculars. And you can see there's an egg mass here on this tree. But here's the one false positive. This is just, I don't know exactly what this is, but it's not an egg mass. If you get multiple angles, you can usually see whether it's raised up, like if there's actual body to the egg mass there. And in this case, this is just really flat to the bark. But it looks pretty convincing to me, and it takes some effort to inspect things to make sure you're not getting false positives. And this is just a picture to show how hard they can be detected, even when they're right in front of you. So this is, depending on the texture of the bark that you're working with, egg masses can be super clear or they can blend in pretty well, like the egg mass here. And there's also some variation in whether egg masses are covered or not. So we get, this is just a cluster with some uncovered eggs and some eggs that have been covered with waxy coating. And that can affect how easy they are to find as well. So we found from our surveys here that egg mass counts are moderately effective at detecting lanternfly presence. So of all the sites where we found nymphs present in the following spring after we did our egg mass counts, we found egg masses in the previous winter there at just 33 of 65 sites, or 50%. So half the time we went, looked for egg masses, found 0 egg masses in a pretty thorough search, I would say, measuring, looking at every stem and documenting for each stem how many egg masses are there. And then, so we recorded a 0 for egg masses, and then when we returned in the spring, we did find lanternfly nymphs present there. So relatively frequently that happened, I think. And then you do better if you include the binoculated search above 3 meters in your assessment for whether there are egg masses there. And so we found egg masses at 63% of the sites where we later found nymphs using binoculars to search high entry. So slightly better, but still far from 100%. One question we had asked when we were setting up to do these egg mass surveys was, how closely does this year's count of old egg masses correlate with the prior year's new egg mass count? So I'm kind of asking, if we look at the old egg masses here, how reliably does that predict how many new egg masses were there? And so if there were a strong correlation between these two things, it might allow us to estimate the population size in a prior year based on the number of old egg masses that we counted. But of course, egg mass deterioration affects not only the presence, whether they're there for us to count or not, but also how easy they are to see. Here's an example of an old egg mass that sticks out pretty clearly here. So they can remain detectable for a while. What we found, and this is kind of a first look at this data, is that the old egg mass counts they do correlate with the number of new egg masses that were present the year prior, but the predictive power there is pretty low. So this shows on the x-axis the number of old egg masses that are present this year, and on the y-axis the number of new egg masses that we saw the year previously when we did the prior survey. So this is one advantage of being able to go to the same plot repeatedly year to year and kind of build these time series of different egg masses. But you can see there's a positive trend. The dotted lines here show a prediction interval, not a confidence interval for the mean. So we predict that 95% of all our egg mass counts would fall within this range. But you can see for a plot where you find 10 old egg masses, for example, there could have been around 10 new egg masses the year previous, or there could have been up to 40 egg masses the year previous. So you're kind of limited in your ability to say how many egg masses were actually laid the year previous based on counting old egg masses this year. So once hatch occurred, we started up with regular visits to these plots, trying other sampling methods. We did timed visual search for nymphs and adults, and we carried the search out in these same plots where the egg mass counts occurred. And we have a 5-minute search. When you get to super high densities of lanternflies, sometimes 5 minutes feels like you don't have quite enough time. So there is some occasions where you might wish for more time, but a lot of the time, especially because a lot of the plots we're looking at very low densities or no lanternflies, 5 minutes feels like a long time to be searching. And so we visited these plots every 2 weeks from May through November. And on each of these occasions, we had 2 observers making independent counts. So taking care not to say, "Ooh, I found one." So getting an independent count and an independent assessment for whether they've detected them or not. And we asked both the observers to cover the entire plot. So we got 2 counts of the same area from independent observers at the same time. And so based on these counts, we can use mixture models to estimate the detection probability and the true abundance of lanternflies in the plot. So these models kind of encapsulate the idea that each count depends on the number of individuals that's truly there, and also on the probability that each one is detected. So we can model with this mixture model, I'll try and get through this quickly, but so we assume the actual number, this big N in the plot, follows a Poisson distribution. The count that we achieve, so this is the number that's actually there in plot I on date J. And then the count that we achieved in plot I on date J by observer K follows this binomial distribution, where you have the number that's there as your number of trials, and then some probability that you detect each one. And then we use a beta distribution for this detection probability. So we say this probability of detecting them depends on the time of year and also the observer. And it has a mean value at this big P. And using the beta distribution lets us account for the fact that there's clusters of lanternfly. So you're not equally likely to find each individual lanternfly. Sometimes you'll look and look and look, find none, and then find a cluster of 5 all at once. While an observer might have looked and looked and looked and found 0 the whole time, just because they missed that cluster. So using this beta distribution lets us account for the fact that they appear in clumps. And then you can predict the mean probability of detecting an individual with a regression here. So you have an intercept and then a term for the day of year. So we were interested in how the probability of detection varies over time. And also we can include a term where different observers have different probabilities of detecting lanternflies. So some people are just better at it than others, I think. That's how it looks based on our stuff so far. So this approach assumes that the population is closed and that there are no double counts or other false positives. And it does require these 2 independent counts. But with this method, you can get a guess for how many are truly present in the plot. So this is for just one plot across time, how many lanternflies are there. And so these black points show each of our observer counts, while the red point shows an estimate for the true abundance in the plot at that time. So you can get some, you can also get confidence intervals around these, but you can get a guess for, based on the counts that we saw, how many are actually there. And you can also get an estimate for detection over time. So this is a plot of across the day of the year within the growing season, just from days from January 1st. What's the probability of detecting a single lanternfly? And so you can see our detection improved over the course of the season, which makes sense because individuals themselves are larger and behave differently throughout the course of the season. So you get improved detection of lanternflies over time. So this is just to find one individual. So you can see if there were just one in your plot, and you look early in the year, there's a good chance you'll miss it. But later in the year, if there's just one in your plot, you do better. You get up to relatively high chance of detecting them. And this is a little bit preliminary. There's some other fiddling with the model that I think we're going to do to try and improve, get to the data, but a rough look at how detection changes through the year. Of course, you do better if there are more individuals in the plot. So this is if there were 5 individuals in the plot, what's the probability that you would detect at least one of them over time? So you can see a big jump already. And then if you get up to 10 individuals in the plot, you can do pretty well, or even when they're small individuals, you can do just for saying yes or no other lanternflies here. What's the probability that you'll see at least one of them? You can do fairly well early on, even when they're very small. So in addition to these visual searches, we also use sticky bands. We use the inward-facing sticky bands manufactured by Bug Barrier. They reduce bycatch compared to the outward-facing sticky bands. And we installed them on Tree of Heaven in our plots if available. And if not, we set a priority for other trees to use on. Based on Joe's talk, I think we definitely should have included black walnut, and maple I think should be earlier up our list here. But that's for next season. I think we'll maybe make that change if we're able to do some sampling this upcoming season. And we check these traps every 2 weeks, June through November in 2020. And then we did it for the whole season this past year, May through November in 2021. And we evaluate these traps by considering the cases where one method detects lanternflies. So if one method said there's lanternflies there, we ask, did this trap also detect lanternflies there? And we analyzed whether the probability of detecting lanternflies was affected by the seasonal timing of observation and the density of lanternflies present at that site. And we do know this method. I'm still working on improving the analysis here because the approach to analysis misses cases when lanternflies are actually present, but all three methods, so our search and each of our traps fails to detect lanternflies. But you can see the bug barrier here, the effectiveness of bug barrier for detecting lanternflies varied over the course of the season. So we've got on the x-axis here the maximum of our 2 counts for lanternfly, the number of individuals in the plot. So just a measure of abundance of lanternflies in the plot. And on the y-axis, the probability of detecting presence. So it makes sense that as you get to a lot of individuals in the plot, you're very likely to detect them. But this animation showing throughout over the course of the season, there's some variability in how likely you are to detect them. So early in the season, you do fairly well detecting them. But once you get to the summer, you drop down to less effective at detecting these very low densities of lanternflies. Another thing we were interested in, and so beyond just saying whether the traps agree with our visual count for yes or no, are there lanternflies there, seeing if they agree in measuring the number of lanternflies there. So this is just a quick look at, so this is the abundance as estimated by our visual count on the x-axis, and the number that we caught in the bug barrier on that date. So there is a strong positive correlation there, a significant positive correlation, but there's a lot of noise as well. And it kind of makes sense because they're measuring 2 different things, really. One is a measure of how many are present in this fixed area, and the other is a measure of over the past 2 weeks, how many lanternflies climbed up this particular tree and got caught in the trap. So I'm working on improving this analysis, taking into account time of year and also the size and species of the trapped tree might affect how closely the agreement between the trap catch and the abundance estimated by visual counts, how closely those line up. We also use circle traps, which you've seen previously in Joe's talk. They're modified plum cuculio traps. They exploit the climbing behavior, so they funnel individuals into a collection bag of trees. And we use the same preference for hosts, and we have 1 trap per plot. And we did switch this past season, switch the trees between, or the traps between trees 2 times throughout the course of the season. And we check these again every 2 weeks. And we find a similar trend where the circle traps effectiveness for detection varied over the season. Maybe slightly lower than bugbear on average, but I don't think there's a real indication that there's just any significant difference between the 2 traps in their ability to detect lanternflies. And this is similar to the plot we saw for bugbear. The visual count and the trap catch and circle traps agreed on for not just whether yes or no they're there, but how many are present in the plot. And you can see again, it's pretty close to the same story that we had for bugbear. It's almost exactly the same story, really, where there's correlation there, but a lot of noise as well. So difficult to say whether they, or how closely they agree. So some thoughts on future directions. We still need to improve our analysis of trap sensitivity using occupancy models to more accurately assess abundance. We're working to conduct, I went through that bit about mixture models and how we use it to compare our duplicated counts for numbers of individuals in plots, but we can do the same thing for egg masses. So we're going to visit more plots this upcoming season and repeat our egg mass counts in these plots so we can get an estimate for the detectability of egg masses that way, and also see if it differs for our counts above and below 3 meters. And then the last plot I showed there is the relationship between trap catch and visual counts, digging in to see whether the tree species that the trap's on or the size of the tree matters, as well as the time of year. And then finally, we'll examine this is kind of a future goal, really, is examine the influence of imperfect detection on validity survey. So does the fact that you miss them 10% of the time when they're present change how you should plan your sampling? So this is more of a modeling question, I think. But with that, I'll say thank you to collaborators and a lot of help in the field.
All right, thanks, Joe. And there's a question in the Q&A that I'll ask you to take a look at, see if you could provide some answers to that. Somebody else might be able to put in some other information on that too. But yeah, thanks, Joe. But it's 11:10, so I'll need to move on to the next presenter. So our next presenter is going to talk about the Verticillium wilt pathogen in Tree of Heaven. And it's Dr. Charu. He's the president of Bioprodex Incorporated. He's also the CEO. And then he is also an emeritus professor of the University of Florida. And if you're ready, Dr. Charu. Yeah, you're muted if you're talking right now.
Oh, okay. Greg, can you hear me?
Yeah, I can hear you now. Thanks.
Okay, very good. I thought I was going at 3 o'clock.
Oh no, it's 11. On the
Okay,
agenda.
that's fine, not a problem.
Yeah.
But one little problem, somebody decided to do some roof repair over my
Okay.
head. Let me ask them to stop.
Okay.
Hello. I'm sorry about that, Greg. I'm
okay.
all ready.
Yep. Okay.
Okay. Are you going to load the presentation?
Yeah, I can ask Jacob to do that. If
Yes,
he's got that.
please.
Jacob,
Yeah.
if you could load the presentation. All right.
There you go. Thank you
All right.
very much.
Sure.
So we are in the second year of our attempt to gather data needed for registration of Verticillium Non-Alfalfa. This is not my effort alone. It's a team effort. And the team consists of Professor Don Davis at Penn State University, Dr. Matt Kassan, who was Don's PhD student, now at West Virginia University, and Anton Bardwan and Dr. Scott Salam at Virginia Tech. And I'm doing all my work at the USDA ARS US Horticultural Research Lab. And Dr. Erin Droskoff, my former student, is the research leader there. And she has kindly allowed me to use her greenhouses and lab facilities for this work. Okay, I think you may have to move the slide.
Yeah, you just ask Jacob to advance it.
Sure.
He'll go ahead.
Okay, so controlling Tree of Heaven by biological means has been a long-standing key objective in the US, especially Forest Service, leading the effort to find agents. The hypothesis now is that with the SLF in the mix of invasives, removing Tree of Heaven or reducing its population density will help towards the management of SLF impacts. Now, next, for those of you who have not heard my talk in the last 2 years, very quickly, Verticillium Non-Alfalfa is a fungus that causes wilt disease on Tree of Heaven. It was discovered around year 2000 in Pennsylvania, and it has been studied by Professor Don Davis and his students at PSU and subsequently at other institutions. This is a native fungus, not an introduced pathogen. In fact, there are reports going back to the early 1900s of verticillium disease in New York City on Tree of Heaven and also in Paris, France. So this fungus has been probably around, and now its presence has become more noticeable and more intensive in its effect. VN, or Verticillium Non-Alfalfa, This is a newly erected species. In other words, it's not newly discovered. It was created out of an older species as a separate species. It is related to Verticillium alboatrum and Verticillium dahliae, two very bothersome plant pathogens that attack several horticultural crops. TOH killed by this fungus has also been found in Virginia, Ohio, and across the ocean in Austria, European Union. The fungus kills TOH of all growth stages. In studies at PSU, it has been found to be relatively host specific to TOH. It has been shown, this study was based on testing plants that occur in the same habitats as TOH. And it was found to attack a couple of other hosts, but I won't go into it right now. Typically, the infected TOH is killed. 100% kill is seen in some studies. It has naturally killed thousands of TOH in Pennsylvania, Virginia, and Ohio. The fungus is easy to culture and produce spores, and it is easy to apply. Now, all of these are very favorable features that should help in the registration process. Suppose it is not a native, we would be in a totally different realm of regulation. So these are all very helpful. Next. Next slide. Okay, some pictures. A healthy tree canopy with a lot of TOH, and another place where a large number of TOH trees have been killed. This is a healthy canopy looking up, and a couple of infected TOH. Leaves are all shed, and basically the plant is starting to wilt. One very interesting feature, which is very helpful for biological control, is that the fungus spreads by natural root grafts of TOH plants. Next. Well, so far all studies have been somewhat limited, but in a large field trial, the fungus was shown to be highly effective across climatic and stand variables in two states, Pennsylvania and Virginia. This study was the PhD work of Rachel Brooks and colleagues in Virginia Tech and West Virginia University. This is a 3-year field inoculation study done at 13 sites across 4 hardiness zones in Pennsylvania and Virginia. It included 3,245 TOH trees. A Virginia isolate of the fungus was used. VNAA 200, because the Pennsylvania isolate, which we are going to develop, could not be used due to state not allowing the fungus to be imported into Virginia. The results indicated with slight variations correlated with climate and stand variables. This Virginia isolate always caused severe disease and some tree death during the 3-year study period. The disease spread quickly to other TOH trees in the forested plots. Next slide. This map shows that the locations where the trials were done. The circles indicate new sites chosen for the study, and the triangles indicate sites where previously the disease had been found. Next slide. This is over time, 0 months after inoculation, 3 months, 15 months, and 27 months after inoculation. These are pictures looking up with the GoPro camera, looking at the canopy. to see how the fungus is performing. This series was inoculated with Verticillium Non-Alfalfa alone. This series was inoculated with Verticillium dahliae. I'll come to this fungus one more time later. Here, both fungi were used, and this is control. As you can see, as the time progressed, during the nearly 2-month period, 2-year period, the non-alfalfa inoculated plants started losing their leaves and started to die. When both fungi were used, there was some similar results, but I won't call it exceptionally synergistic reaction between the two fungi. And Verticillium dahliae alone did not provide that great of a control. These are control plants. Next slide. So where are we in the registration status? Okay, going back to the previous slide to summarize that. So the overall conclusion was this fungus, at least within that mid-Atlantic area, would perform well across the different hardiness zones and possibly slight variations in the climate. This is good news. And registration status, we had a meeting with the EPA, what is called a pre-registration meeting almost 3 years ago. And at this meeting, we laid out all the published information, and EPA reviewed all of that and expressed no reservations about possibly registering the fungus. So in other words, EPA is open to registering this fungus. There are fungi which EPA would have immediately thrown up red flags. For example, fusariums produce mycotoxins, and I know, but from experience, there's no point in going to EPA with some of those wilt-causing fungi. Now, EPA will accept all published data as part of the registration data package. This will save significantly time and cost. What are the aspects they will accept from the published literature? Taxonomy and species designation, proof of concept that a Pennsylvania isolate VNAA 140 is a promising biocontrol agent for TOH, and there is diagnostic sequences available for this isolate and many other isolates. This is published. Now, this is an important data issue. We don't have to worry about it. It's already there now. So we have designated this Pennsylvania isolate as the candidate for registration. The registration is going to be given on a particular isolate or strain. So this will be that isolate. EPA then stipulated a list of required empirical data it needs to assess the registerability of this fungus. Next slide. So these are the supplemental and new data we had to produce. Non-target plant host range study. This is consisting of important horticultural crops, grapes, fruit and nut trees, and representative environmental species. Then the question is, how does this fungus survive and spread in the field? So we are doing an inoculum dispersal and spread studies. Efficacy trials with the bioherbicide formulation that we are close to choosing for registration. This will be done in greenhouse trials and then in field trials of less than 10 acres. And then if EPA says we need to do an experimental use trial, then we can do it on a much larger scale on areas, total area specified and the location specified by EPA. At this point, I would think that we will go to the respective states where we will do EUP studies to get permission, but I think EPA, EUP will probably override some of the states considerations, but I'm not an expert on this. But I'm just saying that EUP will have give us the access to do the studies in several states with this Pennsylvania isolate. Non-target, non-plant pathogenicity, toxicity, hypersensitivity in all these studies. These have to be done by certified labs, and we are close to getting this started. And this coming fiscal year, we'll start honeybee studies. This is an important consideration EPA always asks because of the concern about honeybee the population decline and all that. Next slide. Where are we in data gathering? Ongoing is non-target plant host range studies with the Pennsylvania isolate on crop and native species in Fort Pierce, Florida. Screening of the Pennsylvania isolate on mature, field-grown grape, apple, and peach trees. Dispersal and persistence of this isolate in soil. And efficacy of the Pennsylvania isolate in Tree of Heaven stands in Virginia. This has not been done. The study I referred to by Rachel Brooks, that was, as you remember, is a Virginia isolate. So this study will look at the Pennsylvania isolate and see whether it also performs the same way. The population structure and virulence of Verticillium Non-Alfalfa isolate from a fairly large area in mid-Atlantic states. This is being done by Donald Davis. And whatever we get in our collection, the genomics of these isolates as compared to the sequences of the Pennsylvania isolate chosen for registration. This is being done by Mac Kassan. And large-scale production of this fungus has already been done. And this work is done by Sylvan Bio with their own funding. And Dr. Mark Watt is the leader of this work. Next slide. So what have we done with host range study? We've been at this since the fall of 2020. So far, we have tested 74 species in 44 genera and 26 families. This is all done in a greenhouse according to APHIS PPQ permit rules. We have used seedlings that we raised from seed or saplings we bought from nurseries. These test plants are inoculated by dipping their root mass for 30 minutes in a suspension of 1 to 10 to the power of 7 spores in water. This is the inoculated treatment. Sterile treatment is just sterile water. And then after 30 minutes, take these plants and plant them in potting soil and pots. The plants are kept for 3 to 5 weeks in the greenhouse and rated for disease development or lack of disease. And if the disease is present, the symptoms are noted. TOH is included as a positive check for fungal infectivity in each batch of experiment. The experiment is rejected and the results not considered for use if TOH developed no symptoms. So we have had a couple of these costing us each time about 5 to 6 weeks of our time. Mind you, the greenhouse has only limited space, so we need to do this in several batches. Good news is, so far, no non-target species has been found to be susceptible to the fungus. Next slide. Okay, these are the symptoms we are looking for on TOH. Either you get patches of chlorosis or what is known as vein clearing. That's not quite clear. Chlorosis along the veins. And a little bit of that is seen here. And of course, we would like to see plants wilting and dying like this. So we see chlorotic leaves and some vein clearing, leaf desiccation and leaf abscission quite commonly. plant death has been somewhat rare. And we think it is because the temperatures in Florida are much higher than in mid-Atlantic states. And the temperature does have an effect on the efficacy of the fungus. We are looking at this because this will have an impact on where the fungus might not work at all, such as very hot Southern California. It's probably not going to work in Indio, California. Next slide. Host strain studies on grape and apple. Anton Bardwan and Scott Salam, they are doing this work in Virginia. Established grapevines in a vineyard, Crimson Cabernet, Chardonnay, Cabernet Franc, is a stem injected with the isolate spores, monitored for symptoms over 2 months. And the result is the fungus was not re-isolated from inoculated grapevines, and no disease was evident on any of the varieties at any time. And then trees in an apple orchard that were planted in 1990, so they are mature old trees, were inoculated, 6 for inoculated treatment and 6 for control. And again, observed for 2 months. Here again, no verticillium symptoms were observed, and the fungus was not re-isolated from inoculated apples. The TOH plants, of course, showed the disease. Next slide. The population studies is ongoing at both Penn State and West Virginia University. Don and Matt have made a collection of Verticillium Non-Alfalfa isolate from these 3 states. What is ongoing now is with these isolates is population genetics, phylogenetics, and inoculation studies. Assess the diversity of the isolates in this population. For genetic analysis, 2 methods are used: microsatellite markers and genotype by sequencing data. Virulence and specificity of isolates to TOH is also determined along the way. Out of this, a molecular data set will be developed to compare isolate that make up this population. Why is this important? Well, essentially, it assures us what we are dealing with. Whether we have a fairly homogeneous population, or are there mutants all over the place, which could raise questions on the part of EPA. So this data set will be very useful. Next slide. So here are some other interesting points that I must mention. Now, on the first day, we heard a very interesting talk on plant grape viruses that might be transmitted by SLF. Well, can SLF transmit Verticillium Non-Alfalfa? If it does, it'll probably help the biocontrol effort. So Brooks et al. I thought I turned off the phone. Brooks et al. Tested whether SLF could spread the fungus from symptomatic fungus inoculated TOH to healthy TOH in controlled lab settings. The insects were allowed to reside, feed on inoculated TOH, and then transferred to healthy TOH. The study included monitoring TOH seedlings for symptoms and sampling seedlings and SLF for the presence of the fungus. So there was no evidence that transmission took place. So SLF is unlikely to help spread the fungus in the field. But as in other wilt diseases, any weakening of the TOH plants by SLF infestation might promote and enhance infection by Verticillium Non-Alfalfa. So in effect, there will be an indirect effect of SLF feeding on the efficacy of the fungus, I hope. Next slide. Now, Verticillium dahliae, as I said, is a problematic species found infecting probably a few hundred species. It is more common plant pathogen than Verticillium Non-Alfalfa. It infects and causes wilt disease of TOH in several locations in the US and EU. Often it is sympatric with Verticillium Non-Alfalfa. For example, Matt Kassan reports to me that in West Virginia, he's finding more Verticillium dahliae on TOH than Verticillium Non-Alfalfa. However, dahliae is not as virulent or aggressive on TOH as Verticillium Non-Alfalfa. Excuse me, 1 second, please. Co-infection of Verticillium Non-Alfalfa and dahliae does not cross-protect TOH against VN. This is something I suspected might happen, but it did not. So this is good news. But at the same time, the combination co-infection does not also enhance Verticillium disease. If it does, then it'll be good. In any case, we will not promote the use of dahliae due to its broad host range. Next slide. So plans for this year is to continue gathering all these data, overall biology and safety data, complete the development of a prospective commercial formulation, which is almost done now, and start the toxicology studies, which I didn't put here, and submit whatever data we have on hand to EPA in 2023. Next slide. So that's it. So again, thanks to APHIS for the PPA 7721 grant, and also USDA Forest Service Eastern Region and State and Private Forestry, which gave us a 2-year grant. So this is where we are. And so what's the prognosis? So far, all the data indicate, and EPA posture indicates, that we should not have any major trouble in getting this registered. But then gathering the data has been somewhat slow. Number one, due to the pandemic, the USDA lab had very severe worker schedules were limited. And the number of people in a lab at any time that was limited. So these things slowed things down quite a bit, and the temperature effect is somewhat troublesome. And as I said, the fungus will probably work in all the places where there's no tropical like climate. Matt Kassan, from his experience, tells me that once the fungus is established in the plant, during hot months, it just stays there. And when the cooler weather comes, it starts wilting and killing the plant. So this is something we'll be watching and trying to adjust our strategy.
Okay.
So I'll stop there, and I'll be glad to answer any questions.
Yeah. All right. Thanks, Dr. Charu. Yeah, we're a little bit past on our agenda, so there are quite a few questions for you in the Q&A. If you see that down at the bottom, if you could go to those and provide answers, that'd be great. But,
sure, I'll do that. Thanks.
All right.
Thank you, Greg,
for your
and
presentation. Yeah.
Thank you, Jacob.